Okay. Okay, there we go.
Excellent.
All right, there we go. Okay, so I'm just going to briefly introduce Ben Hampstead. I think many of you know Ben. Ben is the Brent Professor of Psychology here in psychiatry and um, has a staff position at the VA as well. Ben is also the clinical core leader and neuroimaging co-core leader for the Alzheimer's Center here, which is how I met him. Ben has been extremely effective in these particular roles and also has developed a very robust um, research program focused on using neuromodulation in a variety of settings as part of his primary research. And so he's going to talk to us today about neuromodulation, Parkinson's, and Lewy body dementia. Ben, thanks very much for presenting.
Yeah, no, thanks so much for having me. And I think that seeing the folks in this session here helps me to kind of know that I can focus on a little bit more detail than sometimes I otherwise do. I think that I've given a number of talks on campus lately, so I suspect that some of this will be somewhat familiar to some of you. But I think we also tried to throw in some, I think what will be an intriguing jumping off point using some case studies and case series to start to ask the question about how should we or how can we use some of these non-invasive measures in some of the patient populations to both better characterize the sample as well as to consider treatment interventions or interventions for them. So with that, again, the obligatory conflicts disclosures really don't have anything that's that directly relevant here, aside from the potential headgear that we've licensed and have a patent kind of pending under review for. Again, the main focus of my work is, again, knowing all of you or many of you, the idea here is that we know that the neurologic injuries and diseases affect cognition, emotion, and everyday functioning. And that has then downstream effects with the family and others involved in the patient's life. We know at this point that medications often do very little to address cognitive functioning. And so the idea here is that we really want to look at different non-pharm treatment options. And I think that traditionally it's kind of been viewed in a dichotomous approach that either you can have non-pharm or you can have pharm. And if you have one or the other, you don't need the other one. I think that's, hopefully in the past, I think that we have a range of options and we should really be considering how best to use them to address the needs of the patient. And, again, just coming from the Alzheimer's realm where aducanumab looks like it may potentially clear the disease pathology, but not alter clinical status, I think that's a perfect example that if, future disease modifying medications can reduce that downward trajectory, that now we have a different platform to really try and address patient level needs. So, again, I'll kind of focus on a couple of the non-invasive brain stimulation approaches. I'll show you some evidence that seems like some of these techniques may enhance our characterization and seem to align with, I think, some of the themes that Roger, if I recall, the focus of the Udall Center on cholinergic pathways. I think there's some interesting evidence kind of supporting at least TMS-based characterization. And then, again, using some of these case studies, how can we translate some of the diagnostic, especially the imaging findings that we have, into actionable treatment plans? And, again, I'm happy to chat a little bit more about some of the newer methods that they can focus perhaps a little bit more than some of the existing methods. A few caveats. Again, I've pulled in some of the case study case series data because although we had planned to be further ahead, I think COVID has really affected us getting people in as it has so many others. But I think one of the big areas that our research program is really focusing on are these fundamental lack of treatment parameters. So how much, how long, who should we be giving this to? Where do we target? Should we take a uniform approach, one size fits all, an individualized approach, or then some combination of network-based but individualizing it? And then if we think about cognition, When should we be applying this stimulation? Should we be doing it before to kind of prime the system? Should we be pairing stimulation with, say, a cognitive training? Or should we be doing stimulation after and kind of rely on residual cognitive after effects that we know can affect things like resting state fMRI connectivity? So a lot of questions, not a lot of answers, unfortunately. From a safety and tolerability standpoint, there's very little evidence to suggest that the techniques that I'm talking about, the especially direct current stimulation or TMS, transcranial magnetic stimulation, have any significant safety related issues. I think that the risk of seizure, even for TMS, even in those who have epilepsy, is still at about 1%. So pretty low. From TDCS, to my knowledge, there's not been any reported seizures caused by TDCS. There were a couple of studies that I did find with those who have seizures where the TDCS may actually suppress the amount of seizure activity and the frequency of seizures. So I think the question really comes down to more of tolerability. And again, I'm happy to talk a little bit about that because we have a good bit of data there as well. There are a lot of different types of hardware, and so I'll go through a few of these in passing, but again, happy to talk a little bit more and to give people a sense of some of the stuff that we're doing to try and get at least the multi-electrode TDCS in patient hands out in the home so we can do remote sessions. And I think that's really an intriguing option for us because we've been able to deliver stimulation across the state of Michigan as part of our research studies. So at least addressing the COVID-related challenges has yielded some interesting advances there. So again, I know this group, I know that many of you are very familiar with this, so I'm not going to go into too much detail here. But I think that thinking about the non-invasive brain stimulation, transcranial magnetic stimulation, which I'll cover in a minute, and then we have the electrical stimulation, transcranial electrical stimulation, which encompasses these three different techniques that are really characterized by the waveforms that we're applying. And, we can alter those waveforms typically in amplitude, certainly, but then things like ACS and RNS, we can alter the frequency as well. And so I think more and more groups are starting to use things like alternating current stimulation in combination with EEG to try and entrain neural oscillations. So some newer techniques that, again, I won't really touch on, but there's some really cool work with ultrasound and single and multi-transducer arrays that can target deeper brain structures. I'm aware of some work using various forms of laser or various light wavelengths to try and enhance functioning. So again, I know all of you know this, but I always like to emphasize that we are not, TES is not defined by ECT. Very, very different amounts, amplitudes of current, and the last thing we want to do is cause a seizure. So with that background in mind, thinking a little bit about TMS. And so again, I know many of you know this, but I'll kind of go through it again. The most typical way, or kind of the fundamental bedrock of TMS, is to really use motor evoked potentials and this motor threshold. So you basically hit the hand knob of the motor cortex, and then in the contralateral hand, you're measuring the motor evoked potential, so the electrical signal resulting from this pulse over the hand knob. And then you're able to establish the motor threshold. And you can do this either as a resting motor threshold or as an active motor threshold. I think more often than not, resting motor threshold is kind of the gold standard. And so that enables, this fundamental principle allows you to dose. So you can have super threshold. So if you find that resting motor threshold, you can set the stimulator to provide a pulse that will actually cause neurons to fire, or you can have it be sub-threshold, and a lot of studies do, for example, 80% of whatever that threshold was to have a sub-threshold delivery. So there are a lot of different approaches, and I know many of you are familiar with Mike Vesia, who's a great colleague over in kinesiology, who's doing work, I think, actually maybe with some of you already. And so this is from a chapter that Mike and I had put together, just kind of talking about the different approaches. using single and paired pulse, as well as then repetitive TMS. So the RTMS you can do typical, traditional has been low frequency or high frequency stimulation, which can have inhibitory or facilitatory or excitatory effects, with the high frequency being the excitatory. And then newer techniques that can really dramatically shorten the amount of time needed in order to have these inhibitory or excitatory effects. So that theta burst stimulation, where if we give continuous theta burst, then it's a prolonged inhibitory effect, whereas then intermittent theta burst is actually excitatory. And so based on the timing of these single or multiple pulses, you can, as I mentioned, have excitatory and inhibitory. I think some of the ones that we want to focus on are just these things that the SICI, the LICI, the ICF, and the SAI, I think, is especially interesting for some of the data that I'll show you in a minute that come from a pretty comprehensive review, suggesting that the SAI is basically you pair stimulation with a sensory pulse. So you give them this sensory pulse, typically on the wrist, it travels up the spine. And then if you hit a pulse within a certain amount of time after that sensory stimulation is provided, you can actually get an inhibitory effect on this motor evoked potential. And this is thought to be due to cholinergic activity. So I'll show you some data that this actually looks like it's the best measure, or at least a very sensitive measure, in those with Parkinson's and Lewy body type dementia. So just as an example, when these techniques have been applied to those with dementia of the Alzheimer's type or with biomarker confirmed AD, this study had 60 patients with DAT or biologically confirmed AD and 30 controls. And generally speaking, patients with AD showed reduced motor threshold, increased SAI, so greater inhibitory effects, and less LTP-like plasticity, which is measured using intermittent theta burst over the motor cortex and then using MEPs to evaluate that effect. So typically in healthy controls, you see this prolonged excitatory effect, whereas in AD patients, you do not, which is interesting to think about from the standpoint that really we don't think of primary motor cortex being affected by primary disease pathology in AD, at least not to the same extent as other areas. There were no differences in a couple of these other measures. So GABAergic or glutamatergic measures didn't seem to be any different between patients and controls. And interestingly, I mentioned that LTP-like plasticity caused by intermittent theta burst. The area under the curve was actually wonderful at dissociating patients from controls, whereas other measures were, a little bit less robust. And the LTP-like plasticity was unrelated to age, sex, APOE status, or CSF-based AB142, whereas it was related to CSF total tau and p-tau measures. This LTP-like plasticity was also kind of nicely related to performance on a verbal memory test. And those with more LTP-like plasticity had a lower probability of subsequent disease progression. So, I think this, again, is some nice evidence, at least from the AD realm. Now, this I thought was a really well done review that came out last year that looked at all of these different measures across different types of dementia. Now, again, keep in mind that some of the older studies didn't have biomarkers to go on. And so I think we have to kind of, a lot of these rely on the clinical phenotype that now, as especially plasma-based markers come along, we may be able to kind of reevaluate these effects. But at least in Alzheimer's disease, a couple of things emerge. So the green dots are no difference between patients and controls. The up arrow or the down triangle are increases or decreases respectively. So a couple of things really stand out in DAT, where you can see that the resting motor threshold, especially more lately, earlier studies were not as consistent, but more recent studies do show that there's a reduction in the motor threshold. that there's a reduction in the SAI, so that cholinergic path, and then also in that LTP, so that the handful of studies that have looked at it have found a pretty consistent trend of reduction. Now in MCI, oh, it looks like that table doesn't show, but basically the only thing, and only a handful of studies have looked at MCI, and so I apologize that figure is not showing, but really the LTP was the thing that really stood out as being reduced in those with MCI. And again, if we think about the traditional model that MCI is kind of a catch-all in some ways and could have many different etiologies, that is interesting. And some of the biomarker confirmed MCI studies were showing a reduction in LTP. So in thinking about then Lewy body dementia, again, the SAI seems to be one of the more promising approaches, but again, only a handful of studies with a pretty small number of participants in there. In Parkinson's disease, again, relatively small number of studies, but SAI, generally speaking, seems like it's a pretty good marker in those with Parkinson's. And so I thought, and then also PSP actually shifted over to the SICI, which is believed to be a measure of GABA A dysfunction. So these data do raise the possibility that we may be able to enhance our characterization using, non-invasive measures. And this was, I thought, an interesting figure that kind of looked at these different measures. And the closer to the perimeter of the circle these dots are, the more of the studies that have been done in this population showed a difference between patients and controls. So basically in those with AD, as we pointed out, the resting motor threshold, over 75% of the studies have found a difference, whereas those with FTD, really there isn't any difference. And the handful of studies that were done in DLB really didn't show a difference, whereas some of these other measures do look a lot more promising with this SAI, the cholinergic measure, seeming like it could have some promise there. Okay. So I'm not going to go into too much detail on all the treatment studies because the approach, the outcome measure, and the patients do differ so dramatically across these. But I think a couple of things that I will highlight. Prefrontal cortex, everybody loves it. That seems to be the only place that we should be targeting, generally speaking. A little bit of sarcasm there intended, but I mean, if you look at the number of studies targeting dorsolateral or ventrolateral prefrontal, it far outweighs any other brain areas. And I think that would be an interesting discussion to have is, should we really be putting all of our eggs in the dorsolateral prefrontal basket, or are there many other areas that could be beneficial? And I think I have a couple of case studies, Nico, from the patients that we've had a chance to run that I think will be an interesting point of discussion here. So, with that, I think that, I've just moved into some new space here. We have a couple of different TMS units that we're hoping to be able to start characterizing some of the patients that are coming through the Alzheimer's Center. So I do wonder if there's an opportunity to also add in some of the patients coming through the Udall Center so that we can think about cross-center and integrating my research program characterization of some of these participants. So something that, certainly could be pretty interesting for us to think about. Now I'm going to shift gears and talk a little bit about TDCS, or actually a good bit about TDCS, but the basic premise, and again, I know many of you know this, so I'm going to move fairly quickly through it, but the basic idea is that you have two types of electrodes, an anode and a cathode. The anode inserts the current into the head, the cathode pulls it out. So whatever goes in must come out. The traditional approach is to use these big pads. Typically, they're 5 centimeters by 5 centimeters or 7 by 5 centimeter pad. And this is a constant waveform. So once you ramp up, you deliver the amount of current at, say, in this case, 1 milliamp for the desired period of time. Typically 20 minutes is used, and then you shut it off and the current is done. So it's on or off. And so the idea is typically that the current delivered under the anode depolarizes the underlying neurons, making them more likely to fire. Whereas then the thought is that the cathode hyperpolarizes. And I think this is, again, an interesting area of discussion that I say possible hyperpolarizing because I think it really depends on whether on a number of factors that we can discuss more in detail, but that's the basic premise of TDCS. Now, these big pads are fine for saturating large areas, right? But what the finite element modeling is really suggesting is that they're delivering stimulation to a huge swath of the brain. And so if we're doing, say, our traditional, say, F3, F4 bilateral dorsolateral prefrontal montage, you can really see that the bulk of the current is being delivered between these two electrodes and not under it. So even though we may be centering these electrodes over, say, dorsolateral prefrontal cortex, if we define that as, say, middle frontal gyrus, the bulk of the current is really flowing between them and on the medial edges of these electrodes. So we're really getting a lot of off-target delivery. And one of the things that I do think is interesting is, again, I alluded to some of the challenges with the cathode that I'll come back to in a moment. So we're really not targeting where we think we are when we're using the big pads, which is why I really like the quote unquote high definition approach, or as you'll see later, we're doing what I think is multi-electrode. And so these are very far smaller electrodes that then what we can do is say that this is a center anode putting, say, 2 milliamps of current in. We can set the boundaries and have 4 cathodes here that then collect the current and keep it more constrained to the area of interest. And in this is a montage that we used in one of our studies to target the inferior frontal gyrus. And you can see that we do that pretty effectively here. Get relatively little off-target delivery relative to the big pads. So most of our knowledge about TDCS effects come from the motor system. And again, the idea there is that you do TMS, you get the motor evoked potential, then you administer TDCS at some amplitude for some duration using some montage, and then you reevaluate the effect on the MEPs. And basically, the more, the better, or at least the more, the larger the effect under the anode, and that this persists. Newer data suggests that it can persist over 24 hours from a single session. So you get under the anode, you get this excitatory effect where the same amount of stimulation delivered at baseline, if you deliver that same amount of stimulation after TDCS, you get an enhancing MEP, whereas then under the cathode at 1 milliamp, you get a suppressing effect. Okay, great. So couple of things that I'll just use these, and these are busy figures, but the point is basically any of these colored lines were active stimulation. The black lines are sham stimulation. So you can see the Nitschke and colleagues delivered either 1, 2, or 3 milliamps of the anode over the motor cortex and then looked at the MEP changes the same evening, the next morning, the next night, the next evening. And you can see that there really are some pretty prolonged effects for many of these, I mean, at least 2 hours out, right? And that the anode is pretty much always excitatory. Now, the problem with the cathode is that at 1 milliamp, kind of using I took their figures here that truncated the same design, but you can see that at 1 milliamp, it's pretty much always inhibitory, regardless of how long it's delivered for. Whereas at 2 milliamps, it can be excitatory. And then at 3 milliamps, it's suddenly inhibitory again. And again, that depends a little bit, the duration depends a little bit on how long you administer it for. So the cathode causes all sorts of problems that you can imagine has the potential to confound research. And really, I think where we place it, if we're using that pad, is a big source of variability that we need to take into account. And I think as we think about some of the problems our patients have with cognition, if we're extrapolating from motor cortex, okay, that's a different area of the brain. We know that cognition is active, it's slower, it's subject to practice effects, and it's at least partially dependent on a network-based approach, whereas MEPs are passive, they're rapid, they're easily repeated, and they're hardwired. So if we hit that hand knob with enough juice, we know that muscle is going to twitch. So that's not how cognition works, though. So I think it presents a large range of challenges for us as we think about the cognitive deficits our patients have. Add on to that, what aspects of cognition are we really interested in? And let alone then add in mood and emotional functioning and, pre-existing baseline abilities. So we really need some dose response information for non-motor cortex. And again, that comes back to when should we apply TDCS? And there's this concept of functional targeting that we should pair it with a task that engages those regions or networks or that cognitive ability to essentially capitalize on Hebbian plasticity is the thought. That's still largely an unsupported idea, but I think it's not necessarily unreasonable to think of. So just to kind of give you a sense of some of the data that we've done to um, start to establish some dose response or at least some physiologic effects outside of the motor system, I'll show you a couple of different studies that we've done with this spatial navigation task. And the basic idea here is that we know that we can approach spatial navigation in two ways. And so we developed this task that relies on either allocentric or egocentric processing. And we give people instructions to really have them approach the task using either spatial landmarks and spatial relationships, i.e. allocentric, or a series of egocentric frame of reference navigation. I go left, I go right at a given choice point. So we know that these are also problems that many of our patients have in both Alzheimer's and, especially I know some patients with Lewy body and Parkinson's also have these deficits. So I think it's a fairly ecologically relevant task. Then when we first ran this study with healthy young individuals, we found that compared to egocentric, allocentric was engaging regions that we would expect it to. Greater activation in the parietal cortex, as well as the ventral visual stream and the hippocampus. So with that, we then randomized a series of healthy young individuals, and this was using the old pad-based approach because this was back in my Emory days when this was really the only option available to us. We found that when we randomized participants to either have the anode over the parietal cortex and the cathode over the prefrontal cortex, or the opposite montage, we were able to modulate bold signal in a polarity dependent manner. So when we had the anode over the parietal cortex, we saw a significant increase in parietal activation. Whereas then when we put the anode over the prefrontal cortex in the blue group here, we actually, and I don't show the beta weights, but we actually showed increased bold signal under the anode in the prefrontal cortex. So this was encouraging. And so I see Alex Jordan is online, and so he just put out the results of some connectivity data for a study that we did that randomized healthy older adults and patients with MCI, amnestic MCI, presumably due to the Alzheimer's type. And this was a randomized double-blind crossover design where we would give people 2 sessions and we do stimulation, 20 minutes of TDCS with a center anode of 2 milliamps over the right parietal cortex. And the idea was to really try and enhance the spatial processing. So I'm going to focus really on allocentric processing for this set of data. And then at least 48 hours later, to allow sufficient washout, we had people perform a second session. And so again, it was randomized, double-blinded. We don't know, the participants didn't know if they were getting active or sham, but then we would rush them into the scanner and we'd give them that spatial navigation task. Now, when Alex looked at the connectivity of areas that typically are not affected by Alzheimer's disease, right? So primary motor, somatosensory, and visual networks, we saw that really both allocentric and egocentric, there was no effect of either healthy versus MCI or active versus sham. Okay, that's encouraging. But when we looked at the associative networks, we found no differences in the healthy controls, but we found a restorative effect in network-based connectivity in the patients. And again, it was there for both allocentric and egocentric. So there seemed to be this kind of uniform effect that pushed the patients back up into the healthy control range. So again, that's a single session, but pretty interesting effects, I think. Now, we had some extra time as part of this study in the scanner. So what we decided to do was, thanks to our colleagues over in the fMRI lab, to use MRS. And so we placed some voxels in the targeted region, so this right parietal lobe, and the technologists at the time did the best that they could to center this voxel underneath the center anode. We covaried for deviation and overlap of these voxels, and when we did that, we actually found that there was a significant increase in GABA in this voxel that was in the targeted right parietal cortex as a result of active stimulation. But encouragingly, there were no differences in this control voxel that was in the contralateral hemisphere, so the left prefrontal cortex, that you can see essentially no stimulation was delivered there. So this again suggests that there's a localized effect of stimulation. Now, why did GABA change? I think that's a really fascinating question that I'd love to try and pick your brains and try and unpack. I think there are a few different possibilities. But so I think that we're showing some again, physiologic effects of stimulation. Now, I want to focus a little bit on a couple of studies that have used TDCS with those who have Lewy body dementia. I think these are, in my opinion, more cautionary tales. So this was a study that came out, I guess, already 6 years ago now, where there were 13 patients with Parkinson's and Lewy body grouped together. They underwent one single 20-minute session at 2 milliamps. The montage used a dorsolateral for the anode location, and then the cathode was placed on the right deltoid. And the idea is that they're avoiding these competitive effects or confounding effects of the cathode by putting the cathode on the right deltoid. What this does, and this is a model that we had published in a review paper a few years ago, you can see that the current is really shunted down. And you can see that the amount of current kind of delivered is pretty widespread, but it's hitting ventrolateral prefrontal down into the brainstem there, which makes sense, right? Because the current has to flow out of the body. And so what you can see is that there weren't any, there were a couple of effects here. So they used simple reaction time, complex reaction time, divided attention, and then different visual perceptual tasks. Now, the visual perceptual task didn't really show much in terms, if we look at the effect sizes, which I think is appropriate given the sample size, we see that there are a couple of pretty nice effect sizes that suggest that there may be some signal there. The challenge that I see with things like the visual perceptual tasks, this didn't target areas or networks that we would really expect to be fundamentally involved in visual perception. So, I think that the other question has to come to mind is, did these patients have cognitive deficits on the measures? So how well did the montage align with these cognitive processes? And we know that single session is interesting from a sample, but we also can probably surmise that it's not sufficient in terms of how much is delivered at the brain as well as the duration. And I want to focus a little bit more about how much is delivered at the brain level in just a minute. So this is just another example of a more recent, this was a conference abstract that was published, again, had 10 sessions where they used kind of your standard dorsolateral and then supraorbit. And so this is really what they were stimulating, but you can see that they were measuring a wide range of things that are not necessarily going to change with rostral prefrontal stimulation. So, I think that these are more, to me, good examples of some cautionary tales where we have to really do a better job of matching what we're targeting with the patient level needs and align those better with outcome measures. So one of the big focuses that we've had is moving beyond this one size fits all 2 milliamps at the scalp approach, because this suggests that all of the differences in our own head size and shape, let alone the differences in brain morphology, don't matter. They're assuming that everybody has this perfect young MNI template brain, when in fact our patients do not. And so if we set up a montage that's based on this one size fits all approach, we're probably not going to get the desired effects, or at least we're going to see a heck of a lot of variability based on how much current is reaching the brain, which we have to assume is the active ingredient here. And so we analyzed over 300 brains from my program and from the Alzheimer's Center using a couple of different montages. And the bottom line is really that there was a heck of a lot of variability. And so this person down here, 0, is our reference person. So that person got the least amount of current delivered, and we normalize everybody to them. And so what this suggests is that in order to get the same amount of current that this person got at just 2 milliamps, our reference person would need over 14 milliamps of stimulation. So this is a remarkable and frankly, to me, terrifying example of how much individual differences in the brain can make. And again, this is nice because these were patients and healthy controls, but it really highlights the fact that we need to reconsider our dosing approach. So why does it matter? I'm going to go fairly quickly through this because I want to get to some of the case studies and have time for discussion. But this was a study that we did with 12 cognitively intact individuals that we're currently writing up, where we targeted bilateral parietal cortex. And you can see that there's certainly occipital was affected. And again, this was 3 different sessions. So there was a center anode, a center cathode, or a sham session, which was randomized, double-blinded, and then patients were crossed over. And I'll show you data from the allocentric component of that spatial navigation task that I mentioned. And what we did was use graph theory measures of network connectivity. And so the mean strength relative to how much electrical current was delivered to that network. And we extracted the amount of the electrical field values using the exact same nodes for each network that we did to determine the network connectivity. So we really tried to align apples to apples. I'm going to focus on effect sizes again, because this is a small, I think, kind of proof of principle study. I mean, it was a heck of an effort to pull off, but the ultimate sample sizes are low, and we know that fMRI is a fickle beast. But I think that if we go back to how do we power studies, I wanted to focus on kind of medium and large effect sizes. So when we look at network connectivity change, anode relative to sham, as a function of how much electrical field was delivered to that network, we can see that there's some pretty strong effect sizes for a number of these different networks. So default mode, salience, and singular opercular showed medium to large effect size changes, whereas in the medial temporal lobe network had an inverse relationship. So the more current delivered, the less connected it was. But this differed for the resting scans. So I think this highlights the fact that context matters. And we're going to walk away with different conclusions if we measure during rest versus during task. Now, when we compared cathode to sham, we see generally a different pattern of effects. And when we compare anode to cathode, not surprising, we see some combination of those effects, but some pretty large effect sizes. So this suggests that we need to do a much better job of dosing at the brain level, and that we can really use these individualized models to help us in that approach. And we're not the only group showing this variability. This is colleague Adam Woods down at UF, who showed that the amount of current was related to network connectivity change in the dorsolateral prefrontal cortex, not during the sham session, and that it wasn't there in the ventrolateral. But again, keep in mind, this used that F3, F4, where you can again see that the bulk of the current is delivered between these electrodes. So I want to show you a couple studies from Nico and my R21. And so this study uses or focuses on patients with Lewy body dementia. And thanks to Nico, we're using cholinergic PET scans. And then the idea is that we're going to be able to target the areas of dysfunction based on those cholinergic PET scans in order to see if we're moving the needle at all, both from a neurophysiologic and from a cognitive standpoint. So this first person that we had was an 81-year-old left-handed male with 14 years of education. Interestingly, no PET abnormalities. And so that left us in a little bit of a lurch, right? Because we were going to target those PET abnormalities, but because there weren't any, we decided that, okay, we're using a classic N-back task. So 2 back and zero back conditions. So basically, is this stimulus what you saw 2 items ago? So we're doing that in the fMRI scanner. So we decided to take their structural scan and take a network-based approach where we know this frontal parietal kind of cognitive control working memory network is critical for that. So we developed a montage that stimulated those areas bilaterally in this participant. We did this for 10 sessions, and you can see that the amount of current that we were able to get into the brain was, north of half a volt per meter. So a reasonable amount of current kind of delivered here. Thanks to Alex Jordan, who individualized these. Now, again, these are case study results. So I acknowledge that this is tenuous, but I think it does provide some intriguing evidence that we can kind of consider. And I'll show you that we're kind of replicating these results in a few different populations. So the data that I'll show you, the cooler colors represent a decline from post to pre, so greater activation at baseline during this 2-back condition. And then there really weren't any increases. So what we saw was that bilaterally in the IPS, as well as in that left prefrontal cortex, there was reduction. And what was interesting was that we saw an increase in network-based connectivity, largely with the dorsal attention, some with the ventral attention networks, not necessarily in that frontal parietal, which was a little interesting. But that singulo-opercular network that Nico and I had initially posited was, did show some changes. And these are the top 10% of connectivity during the 2-back condition. Now, I know the classic example is to use 2-back versus 0-back. I think there's some interesting reasons that I focused on 2-back that I'd be happy to discuss. But when we looked at their D prime performance, so the change in just raw D prime, we see a really large change for the 2-back condition post-training relative to pre. And then when we look at the percent of improvement versus baseline, we see a pretty striking improvement in the 2-back condition. Now, the 0-back really didn't change, as you would expect. This participant was pretty much at ceiling level for the 0-back, but the 2-back really did change. So this lends potentially some evidence that maybe this is a sign of increased processing efficiency and increased network efficiency. Now, just dipping into some of their data, it was interesting to see that their JOLO, so the judgment aligned performance, orientation performance, substantially increased pre to post. And one of the other things that I thought was interesting was this clock copy condition that is done. You can see that there is a nice, there's some relative change here. The patient inserted this boundary at baseline, but did not post, which this is exactly what they should be copying. So overall, it looked like maybe visual spatial performance maybe also changed. So again, don't want to put too much stock in a single case. And I think the next case is interesting in combination. I apologize, my effects didn't seem to save, but these are the cholinergic PETs. So these are the areas that showed dysfunction at baseline. So we developed an individualized montage that actually got a lot of current into these targeted areas. And you can see the post changes now are really characterized by predominantly increased activation. And what's interesting is that these are really in areas that we targeted, but largely in the right hemisphere. So you can see that these same areas of cholinergic dysfunction were the areas that were showing increased activation. This was accompanied by an increase, especially, it looks like the dorsal attention network was especially affected and showed increased connectivity with multiple networks, whereas then there was reduction in a few other networks. What's interesting, though, is that the percent of improvement in D prime really was pretty nominal. So I think this is within the realm of noise on this end-back task. Looking at some of their other data, though, interestingly, we do see an increase in JOLO. The clock, I don't know, maybe a little bit better. This is from the MoCA. That the copy that I showed you for the last participant right here doesn't really change. I think we could argue that it's dysfunctional in a different way. And, we could squint our eyes and twist our head to maybe say there's something good or bad here. But what I thought was also interesting is that some of the measures of sustained attention and working memory, so this letter number sequencing, seem to move the needle a little bit, and the verbal memory recall improved a good bit. So how much stock should we put in this? I think we can debate that. I think that, at least there might be some signal here. I think that there's some interesting differences between these two patients as a function of where we targeted and as a function of their baseline cholinergic status that we could talk maybe a little bit about. But just to let you know that this isn't the only population that we're seeing these type of changes in. I'll show you a couple of cases of posterior cortical atrophy that we've been working with. And this first one was, again, these first couple of people were more proof of principle to see if we can move the needle at all in terms of physiology. And so we didn't have any baseline PET data for these folks. So we relied on what does the literature tell us about where tau is typically deposited. This was a fairly advanced participant. And we targeted bilateral occipital cortex for 30 sessions. And you can see the amount of current we got in was north of 1 volt per meter. So bilateral montage for 30 sessions. At that time, our eye tracker was actually working, and you can see that the percent of fixations on target, and we have a pretty simple visual perceptual task, and we quantified the number of fixations that were on target at baseline, at post, and at a 2-month follow-up. And we can really see that the participant was much more accurate afterward, and his fixations were relatively shorter post, and that persisted at follow-up. And the patterns of bold signal were really interesting because we see an increase in bold signal during this basic task, as well as what may turn out to be the parietal eye fields. And in changes in network connectivity, looking at how the visual network is changing, we see some really interesting increases versus reductions for this participant. And when we called them back in at that 2-month follow-up, we saw a loss of, kind of a regression toward baseline in these areas, as well as, I think we could question, is this just single session noise? Alex has done a great job of trying to remove any confounding effects. So maybe it's just the system kind of adapting, because we did apply the 30 sessions over a period of about 6 to 8 weeks. And these connectivity changes that were there seem to go away. So these are the changes from post-training to follow-up. And so you can see that there's a reduction in connectivity of the visual cortex to many of these same areas, as well as then basically a restoration of the baseline patterns of connectivity. Likewise, so this was the second patient that we did have clinical FDG PET with that showed a right occipital parietal hypometabolism. So we developed a montage that targeted that. Again, a good amount of current delivered for, again, 30 sessions. We saw an increase in bold signal during that same task. Unfortunately, our eye tracker would no longer work with the U of M system updates. So we've ordered a new eye tracker and currently have that up and running. And we saw a different pattern of connectivity change. Now, this patient, relative to the last, was far less clinically advanced. And we know that we had, that we, at least based on FDG from 2 years ago, we're targeting an area of dysfunction, whereas we had to make that assumption with the previous person. And just as with that previous person, we saw that these changes reverted largely to baseline at a 1-month follow-up. So we're currently doing a pilot study with Bob Kepi and others using FDG in these patients with PCA, and we're doing essentially individualized treatment planning, where we're targeting the area of primary dysfunction for each of these participants. We're printing off headgear that I mentioned earlier that will allow us to do this in a much more rapid, and in at least 2 or 3 of these cases, we're going to train the spouse to do the stimulation remotely, which allows us to deliver stimulation from a lot further away. So with that, I do want to leave a few minutes for conversation, and happy to answer any questions and maybe come back to some of these points that I've brought up. But thanks again for inviting me.
Having trouble. There we go. Okay, so we're open. Ben, thanks very much. We're open for questions. Just I have to ask, so is there any direct evidence that these connectivity measures would actually measure something clinically meaningful?
Yeah, that's a great question, Roger. And I know Cindy, I see you have your hand up, and I know there are a number of other card-carrying imagers in here, so I'll ask y'all to chime in. I think that that's a debatable question. So, for example, with the Alzheimer's Center data, Roger, we're comparing task-based, an associative memory task, and resting state connectivity, and we're finding that the task-based actually predicts the MoCA, as well as composite measures of memory, and whereas the resting state does not. So I think that that's an interesting question that I think you can find data in either direction at this point, to be honest.
Okay. Well, I think Cindy has a question, and then Luis Hernandez has a question. So, Cindy?
Thanks, been a really interesting talk. I'm also interested in hearing from your student later on this summer. I have some reservations about some of the, ones where you have the repeated testing, about how much of that the TD. I'm sure you guys have thought about that, but I think it's a good place to start. Since Alex is working with you, I'm sure you guys have also had this conversation, but I'm just kind of wondering about also taking a look at this and seeing if you can push around the crunch curve, because that kind of working memory task, and the curve that they've shown there is really nice in terms of getting something that's very nice, easy to parameterize, so that way you can parameterize things both on the dose end and on the response measurement end.
I love that, Cindy. Thank you. I think that that's one of the things that I'm really excited about to have more connections over in psychology now, because to be honest, like I mentioned, this was initially, kind of looking at the literature. There's so little that I think is done rigorously. And again, I try and avoid drinking the Kool-Aid with all of this. So let me say that. And, and I fully recognize the challenges that we have with, like you said, the repeated evaluations. But Alex and I have talked a lot about integrating his methods and that crunch curve, because I agree. I think that's such a beautiful example that we can use. And so I think that this is a real opportunity for us to, we seem to have some consistent moving of the needle. How do we optimize our measures? How do we refine the treatment parameters? And I'd love to have more of these conversations, because I think these are the things that we really need to do in order to kind of move this forward.
Great, thanks.
Okay, well, Luis Hernandez asks, can your HD montage be taken into the MRI scanner?
Yeah, can it or should it? So I think, Luis, that the challenges of those are silver chloride electrodes, and they're circles. And so but we do have, I think we'd have to look at it, because I can't remember if anyone has taken them in. Some things that people have done, Minzer in Germany has basically taken rubber electrodes, cut them to the size of these HD silver chloride, and because they're rubber, has put then electrode paste underneath and put them on, and then he created a large ring electrode to serve as the cathode, so that you could have that same general approach. So I think there are a few things like that we could think about that would be perhaps a little more amenable to the fMRI environment. I've also talked with an electrical engineer, materials person up here, and so we're talking about different types of electrodes that may reduce impedance and allow us to get more current in more comfortably with less hassle. So with some of those, I think that those would be MRI compatible. So I again would love to chat with you a little bit, because I know you've been doing a lot of work trying to measure the current flow using various sequences. So
yeah, I'd love to talk more about it and see if we can make it happen. Yeah, long conversation though.
Yeah.
Okay. All right, very good. Any other questions or comments, please? Okay. Well, in that case, we're going to stop, give people back 2 minutes. Thanks very much, Ben. That was a very good, as usual, very interesting talk. And I encourage everybody to talk to Ben about some of these other issues that come up in the questions. Thanks a lot. All right, thanks
Thanks,
everybody.